Introduction to golden rice. So what is golden rice? Golden rice is a variety of rice derived via genetic engineering to biosynthesize beta-carotene. Beta-carotene is a precursor to vitamin A in the nutritional composition of rice. Golden rice is intended to produce fortified food to be harvested and consumed in area with a deficiency of vitamin A diet. Beta-carotene is converted to vitamin A when metabolized by the human body. Vitamin A is required for clear skin, immune responses, and sight. The Golden Rice Project was first established in 1999 when two researchers, Ingo Potericus and Peter Bayer, proposed their project to the Rockefeller Foundation to genetically modify rice for increase its nutrition. The Golden Rice Project began as a potential methodology for vitamin A deficiency. However, despite dealing with laws and regulation governing GMOs, criticism to the project has blocked the development of the Golden Rice Project in emerging regions. The Golden Rice debate consists of two opposing parties. The first one is the oppositions. Friends of the Earth believe that planting golden rice will not solve the vitamin A deficiency crisis. They claim that golden rice produces no enough beta-carotene to cure vitamin A deficiency. About 1.6 microgram per gram of rice and 10% of the recommended intake for vitamin A. The second one is the supporters. The International Rice Research Institute perceives golden rice is a new alternative presented by biotechnology. A clinical trial conducted in 2009 showed the potential for golden rice to have a more favorable bioconversion rate compared to any other biofortified crop. It can provide about 50% of the recommended vitamin A diet from a cup of rice. So, what are the ethical issues raised? Does this risk outweigh the benefits? A consistent ethical framework says that we need to consider whether planting golden rice would cause extensive harm or not. The assessments of the risks and benefits of growing golden rice could provide data whether the Golden Rice Project would have a positive or negative overall impact to the country.
Now, let's go to see the process of creating a genetically modified plant, golden rice. So, these are Agrobacterium tumefaciens. Agrobacterium tumefaciens contains a plasmid, a small circular piece of DNA that has its own origin of replication and is replicated independently of other nuclear material, which is the key to its use in genetically modifying plants. The soil plant pathogenic bacterium has become the most used method for the introduction of foreign genes into plant cells and the subsequent regeneration of transgenic plants. Now let's get to the overview of the process. Roughly, this is how it goes as the picture below. First, the insertion of gene into plasmid using restriction enzyme and DNA ligase. And second, introduction into plant cells in culture that contains tDNA carrying new gene within plant chromosome. And then, regeneration of plant where we soil to grow. Now let's go deep into the process. So in making golden rice, tDNA gene of the gene interest are first selected. So this DNA contained all 3 genes, 2 from daffodils and 1 from bacteria. Phytin synthase and lycopene beta cyclase comes from daffodil, and 1 from Arowina iridophora is phytin desaturase. These genes, along with promoters, are inserted into Agrobacterium tumefaciens plasmids called a tumor-inducing plasmid using restriction enzyme to generate the complementary sequences on the vector and the fragment. And then they join the vector and chromosomal DNA fragment using DNA ligase, so recombinant DNA molecule is produced. After having the vector containing gene of interest, the DNA is transferred into the rice embryo. But how? There are so many methods, but to make golden rice, desiccation was chosen. Desiccation during co-culture dramatically suppressed agrobacterium growth, which is one of the factors known to favor plant cell recovery. It was run out to integrate the vector genes into the rice embryo through direct DNA transfer. So first, these are punched out from a plant leaf and incubated overnight with a culture of vector containing A tumefaciens in urea broth. The discs are then placed in culture plates containing a medium that induces the leaf discs to start growing shoots, which are the precursors of the plant stem and leaves. The dried embryos can be mixed with a nutrient solution containing the recombinant DNA. The DNA should be taken up by the plant cell as the embryos dehydrates, and seedlings can be germinated in the presence of vector containing A tumefaciens in urea broth to assess the incorporation. After a couple of days, these discs are transferred to plates which contain antibiotics corresponding to the antibiotic-resistant microgenes placed in the vector. Only those plant cells that have integrated DNA from the vector will have antibiotic resistance. The medium therefore selects for the transformed plant cells by killing those that do not contain the vector. Once the transformed cells are selected, the growing shoots are transferred to a root-inducing medium where they grow roots and then to soil to grow into transgenic plants. The rice is now grown locally and suited to a particular region's climate and growing conditions. Our golden rice plants are now produced and stored their beta-carotene in their starch.
Have you ever wondered how golden rice helps in vitamin A deficiency syndrome? Golden rice is genetically engineered to produce and store beta-carotene in the endosperm of the tiny plant. So, when a golden rice is consumed, it will be converted into retinol, or also known as vitamin A in the liver. What is beta-carotene actually? It is a red-orange pigment that is usually found in colorful fruits such as in carrot. It has a molecular formula of C14H56. Based on Tim Newton, in his article, he states that the name carotene comes from Latin word "carota", which means carrot. Other than in carrot, beta-carotene can also be obtained from sweet potatoes, pumpkins, and onions. In human body, beta-carotene will be converted into vitamin A, which is retinol. Lack of vitamin A can cause problems related to vision and growth. So, the question is beta-carotene safe to be consumed? The answer is yes. Is there any possibility of overdose? The answer is no. Beta-carotene or golden rice can be ingested in large quantities without being overdosed. Your body will convert beta-carotene to vitamin A as required by your body only. The remainder will be stored in the part of the body or excreted. An example described in the Elsa document mentions that beta-carotene with a dose of up to 20 to 180 mg per day has been used to treat patients with erythropoietic protoporphyria with no evidence of toxicity and without the development of abnormally elevated blood vitamin A. One molecule of beta-carotene can be converted into 2 molecules of vitamin A. It will be broken down in the middle part of its molecule. The oxidation of beta-carotene occurs in the wall of the small intestine, which is the intestinal mucosa, and is catalyzed by the enzyme beta-carotene deoxygenase to form retinol. Later, the retinol was reduced to retinol by retinol dehydrogenase in the intestine. This is the reduction of aldehyde by addition of hydrogen atoms to make the alcohol, which is the retinol. Vitamin A deficiency is usually associated with blindness and growth. Young children in India, Africa, and Southeast Asia who have diets lacking in vitamin A are more at risk of having dry eyes, blindness, or dying corneas. It is also known as white spot. One of the first signs of deficiency is often an inability to produce tears. Other than that, children who do not get enough vitamin A may experience stunted growth. This is because vitamin A is necessary for the proper studies have shown that vitamin A supplements alone or with other nutrients can improve growth. Most of the studies were conducted on children in developing nations. Rice is the most important staple food for hundreds of millions of people in developing countries. Hence, delivery of beta-carotene with the help of golden rice could contribute to a reduction of chronic health problems caused by vitamin A deficiency.
These are the advantages of golden rice. Golden rice contains gene that produces beta-carotene. Beta-carotene provides vitamin A, which helps with eyesight and boosts immune system. It also can help prevent millions deaths and to alleviate the suffering of children and adults afflicted with vitamin A deficiency, which also known as VAD, and micronutrient malnutrition in the developing countries. Only 40 grams consumed daily is expected to prevent death and blindness, with no possibility of overdosing, as the human body only converts the beta-carotene it needs to vitamin A and excretes the rest. It is proof that golden rice is safe to consume and will not cause allergies. These are some disadvantages of golden rice. Golden rice can crossbreed and contaminate wild type. Thus, could create unwanted resistance of pesticides and herbicides, or could increase the strength of wheat. The seeds from genetically modified plants such as golden rice can be very expensive.